Okay, everybody, this is going to be next video, part 2. We're going to look at specifically osmosis and diffusion. Once we look at osmosis and diffusion, we're then going to jump back into a little bit, kind of, again, into the first learning objective about those cellular membrane and some of the transport stuff. So, starting out, I want to talk about diffusion versus osmosis, okay? Biggest thing to remember, the most important thing, whenever we're talking about osmosis, we are talking about movement of water, okay? Nothing else is moving except for water when we specifically define osmosis, okay? Osmosis literally is the movement of water through a semi-permeable membrane. So, for example, we look at this example B right here, where we have a high concentration of stuff mixed into water, so we'll call this a high concentration solution, and a low concentration solution. And this is a semi-permeable membrane. This semi-permeable membrane allows water to cross through, but not whatever is dissolved in, these solutes, okay? So that does is all of this wants to be balanced. The concentrations want to become equal to each other. Remember, because we talked about homeostasis, that dynamic equilibrium, we want to be balanced. So the way that's going to happen is water is going to move across the membrane in order to equal out the concentrations and to create that balance that it wants. That is osmosis, okay? Is anytime we look at water kind of stuff, that we're going to be talking about osmosis. I'm going to jump ahead a little bit because we see an example of osmosis specifically with something else that we really want to make sure that that we cover, because you will get questions on this throughout your career at school. Let me pull up a different document that I know specifically has it in. And this is also something that we have mentioned before. It's about the different types of solutions. We have isotonic, hypertonic, and hypotonic solutions. Let me... There we go. Sorry, it's taking a little bit longer. But this is something that comes back up over and over and over again, and I want to make sure that we look at it. Here we go. So, isotonic, hypertonic, and hypotonic solutions. If you watched my Biochem 1 videos, you know that I'm really big into understanding prefixes, suffixes, and kind of parts of words to help us piece together what's going on if we don't know what else is going on. So let's looking at all of these. Iso, this one right here, prefix right here, iso means the same. So words you're going to see isotonic, isometric, isokinetic, words like this. All these, the second half parts of the words, don't pay too much attention to right now. We care about iso. Iso means that it's the same. It's not changing. Nothing's different. Okay? So, for example, an isotonic solution, that's where the concentration outside of a cell is the same as inside of the cell. So outside and inside are equal concentrations. With this, we get no movement of water. There's no osmosis that occurs in this kind of situation. On the other hand, we have hypertonic and hypotonic solutions. With hypertonic, hyper, think high energy, a lot going on. Hyper can also mean excessive or extra or more. So you'll hear words like hyper, kalemia, hypertension. That hyper, it's high, it's more. Okay, so hypertonic means that there's way more outside than inside of the cell. Because of this, just like the example that we saw before right here, it has a semi-permeable membrane, it's going to want to balance it out. So the water is going to go from that low concentration, it's going to move into the high concentration to balance it out, and we get cell shrinkage. Okay, the cell is going to shrink or crenate. Then the opposite of that, hypotonic, means that the outside of the cell, the outside solution, is going to have a lower concentration than inside of the cell. And again, it tries to balance it. So the cell is going to absorb water, or water is going to move into the cell in order to balance out the concentrations. And that we get swelling of the cell. A trick that I remember for that is a hypotonic solution swells up big like a hippo. That's the way that I remember it. It seems to be the easiest way to remember that for me. Okay, so that's osmosis. Now, talking about diffusion. Diffusion itself is, rather than the water moving, the stuff moves. Okay, the solutes. These solutes are going to move. Okay, so the stuff that's mixed in is what is going to move. So we have simple diffusion. Simple diffusion is where things are just going to move across by themselves without any issues. Okay, this can be done through ion channels, through gated ion channels. This is right here's a little gate. Non-gated ion channels, or it can be just go straight through. Remember we talked about previously how certain things can just pass right through, those lipid soluble things as well as certain gases and such. That is an example of simple diffusion where it just can go straight across. Or if gates or things are open naturally, just kind of chilling, hanging out, we get simple diffusion where it just crosses by itself. One example with water, there are proteins called aquaporins. These are membrane proteins, membrane channels that allow water to pass through. And that's why our membranes are semi-permeable to water. Okay, so that's simple diffusion. Now, it's important to note that a lot of these terms, when we talk about movement and diffusion, these terms can stack onto one another. So we talked about earlier how if we use a membrane protein, an intermembranous protein, that's facilitated diffusion. So if we're using a membrane protein, but we're not using energy, we're not using anything else, that can also be called simple diffusion. Okay, we can call it simple facilitated diffusion. We can just call it simple diffusion. We can call it facilitated diffusion. Any of those terms are fine. Biggest difference is that with simple diffusion, it also includes just things going straight across without a protein. Okay, so if you have a question that involves an intermembrane protein, the best answer is going to be facilitated diffusion. If things are moving across just freely, the best answer is simple diffusion. Now, diffusion in itself, the way that we just described it, where things can just pass through, crossing, according to the gradients and however it wants to try and balance itself out, that is known also as passive transport, which occurs down a concentration gradient. So we're going from an area of high concentration to an area of low concentration of the solute. This is directly through the lipid bilayer, or it uses the channel or the protein carrier, either way. The big thing about passive transport is that it is passive, meaning no energy required. Remember this, no ATP is used for simple diffusion, aka passive transport. Okay? On the other side of the coin, we have active transport. That's literally the opposite of passive transport, where again we can we're going to be using a protein carrier. Some typically not channels, because channels are a little bit harder to control. So we're going to say we're going to use a carrier. We're going to use a protein. It requires energy. It requires the ATP, and it goes against the concentration gradient. Okay, it's super easy to go from high concentration to low concentration. That's what things want to do. So in order to go against that, so another way of saying that is we do low concentration. These little brackets mean concentration. So low concentration to high concentration. Okay? That's what active transport is going to be doing. And we have these carrier proteins. We input energy, and that is going to get us active transport. Now again, remember these can stack. So active transport, because we're going to be involving some kind of carrier protein. Active transport is also facilitated. You can call it active transport, active facilitated transport. You can call it either of those two things, and you're saying essentially the same thing. So even down here, we have that simple diffusion going straight through. We have facilitated diffusion, and we have, so both of all of this is passive transport, and then here it is active transport. And remember, if we're just moving water, that is osmosis. Okay. Don't worry too much about the rate of diffusion, and then we'll get into this next part in the next video. Okay, so hopefully this video helped. Diffusion versus osmosis. Really hit hard. Make sure you know the definitions of stuff, and make sure you know how, like, what is an AKA of what, and how each of these kind of fit together in order to explain what is happening. We'll use a lot of this stuff, a lot of the active transport, diffusion, passive transport definitions in the next couple of units when we start looking at some of the processes that use these. Thanks, and I'll see you in the next one.